Drug design is a complex process that involves identifying and developing compounds that can effectively treat diseases. Over the years, two main approaches have emerged in drug discovery: the classical, phenotypic approach and the modern, target-based approach. This short video aims to compare and contrast these two approaches, highlighting their strengths, limitations, and applications in drug design. The classical approach, also known as the phenotypic approach, focuses on screening compounds for their ability to produce a desired therapeutic effect in a whole organism or cell system. In this approach, researchers start with a disease model and screen a library of compounds to identify those that exhibit the desired therapeutic effect. The compounds are then further optimized to enhance their efficacy, selectivity, and safety. The classical approach allows for the identification of compounds that can modulate complex biological systems, making it useful for diseases with poorly understood mechanisms or multiple targets. By observing the effects of compounds on the whole organism or cell system, the classical approach can uncover new targets and pathways involved in disease processes. The classical approach can identify compounds that interact with multiple targets, potentially leading to synergistic effects and improved therapeutic outcomes. However, the classical approach may identify compounds with desired effects but without a clear understanding of their underlying mechanisms of action. The process of screening and optimizing compounds in the classical approach can be time-consuming and resource-intensive due to the need for extensive biological testing. Since the compounds are tested in whole organisms or cell systems, there is a higher risk of off-target effects, which may lead to unexpected side effects. The classical approach has been successful in the discovery of drugs such as antibiotics, antivirals, and chemotherapeutic agents. It has also been effective in identifying drugs for complex diseases, such as cardiovascular disorders and neurological conditions. The modern approach, also known as the target-based approach, focuses on identifying specific molecular targets involved in disease processes and designing compounds that selectively interact with these targets. This approach relies on a deep understanding of the disease biology and the molecular mechanisms underlying it. The target-based approach allows for the design of compounds that specifically interact with the intended target, increasing the chances of efficacy and reducing off-target effects. By focusing on specific targets, the modern approach provides a better understanding of the underlying disease mechanisms, facilitating rational drug design. The target-based approach enables iterative optimization of compounds based on the structure-activity relationship, leading to improved potency, selectivity, and pharmacokinetic properties. However, the target-based approach may overlook complex interactions and pathways that contribute to disease processes but are not directly targeted. Diseases with multifactorial causes and complex biological systems may be challenging to address solely through the target-based approach. If the selected target is not truly relevant to the disease or its modulation does not lead to the desired therapeutic effect, the target-based approach may fail. The target-based approach has been successful in the development of drugs for diseases with well-defined molecular targets, such as certain cancers, HIV, and autoimmune disorders. It has also been instrumental in the design of drugs that specifically target enzymes, receptors, and other molecular targets involved in disease pathways. While the classical and modern approaches have distinct characteristics, they are not mutually exclusive. In fact, a growing trend in drug design is the integration of both approaches to maximize the chances of success. By combining the strengths of both approaches, researchers can leverage the broad-spectrum activity and phenotypic screening of the classical approach with the mechanistic understanding and precise target engagement of the modern approach. This integrated approach allows for a more comprehensive exploration of disease biology, identification of novel targets, and rational design of compounds. Advancements in technologies such as high-throughput screening, computational modeling, and omics data analysis have facilitated the integration of classical and modern approaches. These tools enable researchers to screen large compound libraries against specific targets, identify compounds with desired phenotypic effects, and understand their mechanisms of action. In some cases, the classical approach can be used as a starting point to identify compounds with desired phenotypic effects, which are then further optimized using target-based approaches. This iterative process allows for the development of compounds with improved selectivity, potency, and safety profiles. The classical, phenotypic, and modern, target-based approaches to drug design offer distinct advantages and limitations. The classical approach allows for the identification of compounds with broad-spectrum activity and the potential to modulate complex biological systems. On the other hand, the modern approach enables precise target engagement and a deeper understanding of disease mechanisms. While each approach has its merits, the integration of both approaches holds promise for more effective and efficient drug discovery. By combining the strengths of phenotypic screening and target-based design, researchers can enhance the chances of identifying compounds with desired therapeutic effects, while also gaining mechanistic insights into disease biology. Ultimately, the choice of approach depends on various factors, including the nature of the disease, available resources, and the level of understanding of disease mechanisms. By continuously refining and integrating these approaches, researchers can drive innovation in drug design and contribute to the development of more effective treatments for a wide range of diseases.